concept of the translation or protein synthesis process. We know very well one amino acid is getting attached to the other amino acid with the help of the amide bond, with the help of the amide bond or peptide bond. So peptide bond or amide bond is very, very important for joining one amino acid along with another amino acid. So then only the amino acid attachment leads to the formation of the protein, right? In the ribosome of a particular living cell. That means dead cells ribosomes will not support the protein formation process. Usually in the case of dead cells, the ribosomes will not remain active for a longer time. And in the case of some cells are formed, for example, if you're talking about xylem fibers or a dead xylem fiber is a dead cell. Phloem fiber is a dead cell. In those cells, we can't find the ribosomes. We can't find the ribosomes. One amino acid joins with the other amino acid by means of peptide bond, right? So now we are coming to the next basic messages. In the translation process, the amino acid is getting attached to the tRNA and making the tRNA as amino acyl tRNA. Can you see this? Amino acyl tRNA, right? So the amino acyl tRNA is formed because of the addition of amino acid to the tRNA, right? So that tRNA brings the amino acid to the ribosome for attaching one amino acid along with the other amino acid, right? So the tRNA brings specific amino acid depends upon the way how the mRNA brings the codon. So I'm the mRNA, I bring specific codon. Based on the codon, the specific type of tRNA brings specific type of amino acid, not randomly. Okay, so you need to understand that the specific mRNA codon brings a specific tRNA amino acid. Why? Because I already told you the first position, this amino acid should supposed to be. The second position, this amino acid should supposed to be. So in each and every position, which amino acid should supposed to be, and how it should be interlinked, how the protein should get manufactured in such a way it can work as an enzyme. Okay, so everything is designed by the mRNA, which takes a message from the DNA, right? So now, if we're talking about the ribosome role, we all know very well the protein biosynthetic machinery ribosome is not present in all the living cells in equal concentration. So what do you mean about that? In the old cell, I'm having less number of ribosomes. I am just formed now only. I'm a young cell. I'm having maximum mitochondria. So I'm a young cell. I'm having maximum mitochondria. I'm an old cell. I'm having less mitochondria. So less mitochondria, maximum mitochondria, moderate mitochondria. Like this, less ribosome, maximum ribosome. Okay, huge ribosomes. So huge ribosomes means that cell multiplies very faster. That cell supports the protein for that cell supports the protein rich organ. For example, skin is protein rich organ. Skin itself an organ, right? So the biggest organ of the body is nothing but a skin, which requires maximum protein. For that to have in system, we need maximum amount of ribosome. So for maximum ribosome, we require maximum amount of ribosomal RNA. Maximum amount of ribosomal RNA, right? So we need to understand this message. The largest subunit site in the ribosomes, the largest subunit site of the ribosome, okay, and small subunit of the ribosome are the two important segments of the ribosomes, right? So we know very well the ribosome skeleton is 23S rRNA in bacteria. Okay, that is this 23S rRNA is acting as a brick of the ribosome. This 23S rRNA also performs or also works as an enzyme, ribozyme. Again, back repeating, 23S rRNA works as an enzyme, ribozyme. 23S rRNA acts as a bridge of the ribosome. So this type of dual behavior sense shown by the 23S rRNA. This ribozyme 23S rRNA is very, very important in bringing the post-transcriptional modification. Post-transcriptional modification means RNA maturation process, right? So now we are entering the next concept called translation. Because of translation, initiation and elongation is the main role. So what do you mean about initiation? And what is the elongation? You know very well. So in the initiation process, the formation of amino acid tRNA is very, very important. The second process of the translation initiation is nothing but amino acid tRNA should enter into the ribosome. Amino acid tRNA should enter into the ribosome. Okay, and then only the translation is initiated. All these things happens with the help of ATP expenses. ATP expresses in the sense the ATP need to get broken. Once the ATP is broken, what will happen is the energy is released. The energy released by the ATP breakage nourishes the protein formation process, right? In the translation termination process, stop codon stops the protein synthesis process. Stop codon stops the or termination codon stops the protein synthesis process in the ribosome. Now, before entering to the lac of codon, we will discuss few questions in the translation and worked examples. First, we'll go for the worked examples. Can you see the worked examples? Give an mRNA sequence. Decodes to identify the amino acid in the resulting polypeptide. The AUG codes for methionine. Okay, GUG codes for serine. So these are the codons codes for these respective amino acids, right? So stop codon signals the termination of the translation. So the polypeptide will be what? Methionine, serine, and glutamine. So these are the three little symbols. This MET, SER, and GLN are all the symbols of the respective amino acids. These respective amino acids are found by the respective codon, right? So now, if you're going for the mutation in the DNA sequence, obviously bring the changes in the polypeptide. If there is a change in the polypeptide are the building blocks of the enzymes. Polypeptides are the building blocks of the membrane. Polypeptides are the building blocks of the peptide hormones. So the peptide hormones are not formed properly, or enzymes are not formed properly, or the membrane structure are not formed properly. This is exclusively because of the change in the DNA. This is exclusively because of the change in the DNA. That is called silent mutation. Silent mutation. See, for example, the GUG, you know it's undergoing changes, it brings the arginine. There is no issue at all. But if the mutation prolongs, okay, the CGA, if it is getting duplicated to two or three in the same place, then there will be issue. So without any interruption in the amino acid sequence arrangement, if the mutation is happening, that we used to call a silent mutation, it won't harm the particular individual who is having this particular sequence. So silent mutation is not an issue. Okay, whereas point mutation is an issue. Okay, that we already discussed with you. Now we are coming for the process of peptide bond formation during translation. We all know very well the peptide bonds are formed because of the addition of two amino acid side groups. Because of the addition of two amino acid side groups, peptide bond is formed. The peptide bond is linking one amino acid along with other amino acid. Like this amino acid is keeping on join with each other and finally making a good quality protein, right? So these are the messages we should supposed to know when you are discussing the third question, the process of peptide bond formation during translation, right? So now, uh, so these are the worked examples. And in all the worked examples, we already told you a number of times. See, can you see this laser properly? All these points are explained point by point. It is not explained as a paragraph. It is not explained rawly. We simplified and we made as a bullet points. This is the way you present it in the answer script. Then you will get the maximum score. Or else, who is giving the clarity, they will secure the maximum score than you. So the way how to present that we explained to you with the worked examples. Whenever you're having time in the Go to University portal, please see the relevant video for the same chapter. Okay, and then we are going for the next important concept in the same translation mechanism. Is how they asked in this number of years about the translation with a lot of worked examples that we need to discuss in detail, right? So, for example, in which particular translation process, uh, the ATP is involved, in which particular translation sub segment GDP is involved, that we need to understand with the questions, right? So, this question is exclusively taken from the 2023 board paper. Okay, given below is the sequence of the bases in the mRNA of the bacterial cell. Identify the amino acid that would be incorporated at the codon position 3 and codon position 5 during the process of translation, right? So, if you are carefully examining the mRNA sequence, okay, they told codon position 3, codon position 5, like this they told. So, we need to start counting from first 3 codon, second 3 codon, third 3 codon, fourth 3 codon, fifth 3 codon. So, like this, we are keeping on counting the third 3 codon. The third 3 codon is UCA. UCA. Okay. which is code for which exactly designs or which exactly governs, which exactly controls the setting, right? The code answer to the question 5 is GWGC, that codes for glycine, right? So here glycine is an optically inactive isomer. So here always your mind should work in dual mind. Okay, here what is this glycine? Glycine is an optically inactive isomer. Among all the amino acids, the only one amino acid which is optically inactive is glycine. So there is a message we need to understand, right? Or understanding is part of that. Okay, this is one of the board question paper we discussed with you. Okay, and now we are coming for the next question discussion. What is the role of the mRNA in the process of translation? Right? What is the role of the mRNA in the process of translation? Okay, any one of you can see this message? Okay, mRNA carries genetic code for the protein synthesis, right? Okay, am I correct? Okay, the sequence of the basis of the mRNA specifies the sequence of the amino acid in this one, right? Now we are coming to the next basic pointers. In the question 3, which of the following is the start codon that initiates the translation? AUG codes for first amino acid in all the protein, right? Methionine. So whenever you're seeing the methionine methyl group is there, your mind should go, "Ine" means what? NH2. So like this, what your mind should work in very behavior. In the sense, okay, here the methionine codes for nothing. Methionine is called by AUG. This methionine is the first amino acid in all the protein, right? So this is the way you are remembering. Why? Because some students are saying methionine codes for protein. No, that is wrong information. Okay, AUG codes for methionine. Methionine constitutes the protein. Okay, being a brick or being a component of the protein, right? So now we are entering into the question number 4. In translation, where does the binding of the tRNA with the mRNA occurs? Okay, we all know very well. Binding of the tRNA with mRNA happens either in the smaller subunit, larger subunit, in the nucleus, or cytoplasm. Of course, in the larger subunit or smaller subunit. Okay. Okay, mRNA. The initial binding of the tRNA with the mRNA occurs during translation in the case of larger subunit. Okay. Basically, this entire event happens not in the nucleus, of course, and not in the cytoplasm, of course, in the largest subunit. Okay. Here, I think so. You all understood. In the case of NCERT example, multiple varieties of questions we discussed. Now we are waiting for the exam. So technology and make it a shorter. For example, you are doing a big paragraph, you break those paragraphs into small sentences and convert those sentences into shorthand. Well, because tomorrow morning I have the board exam, right? I can't read each and every sentence of it. So I have the concise, precise shorthand of mine. I designed this shorthand and a number of time I record a lot of information from this shorthand. Okay, so that shorthand quick revision is very important. So you'll balance the exam properly, right? So this is about the translation. So again, back you need to understand something tricky questions they will ask. In which particular cell of the human being in abnormal condition, the protein manufacturing credential will be observed at the maximum level. Like this, if they're asking, then what type of answer you will be writing? Of course, the tumor cells. Of course, the tumor cells. There is a rare chance for asking these type of questions, but what they will do is the chapter number 8 is nothing but not chapter number 8. Chapter number 7 is celiac disease. The chapter number 5 is molecular basis of inheritance. Both the chapters they intermingle and then they will ask hybrid question. So this probability is also there. So the translation or protein synthesis is maximum in the case of cancer cells, and the translation is very, very slow in the case of the cell which is during the death. Okay, old cell. Okay, or aged cell. These cells can't do the protein synthesis more effectively. Whereas the aged cell or a newborn cell makes a protein at a very faster rate. Okay, can you give one example for what? Yes, of course, we know very well bone making cells, osteoblast, right? Which makes a protein at a very faster rate, right?